Good afternoon. Thank you for coming to our Monday afternoon seminar series. And today I have the pleasure of introducing Mr. Jonathan Lacey. He went for his undergrad, he got his bachelor's in plant biology and with a focus in molecular biology and biochemistry, and with a minor in chemistry, and that was at Southern Illinois University. Before that, actually, he worked at Construction Millwright Incorporated, where he worked on anything that was rotating on a factory line. So he had some experience not in biology before he came here. Currently, his advisor is Dr. Carl Bradley, and with a co-advisor from Dr. Arjun Kashu. And he works on combination of trichoderma and fungicide-treated seeds to control root rot pathogens. So he has spent his summers in Princeton on the Princeton campus, and he's very knowledgeable about everything applied and practical in terms of putting seeds into the ground. And whenever I have questions, I always go about that kind of stuff. I always answer. And without further ado, let's start the presentation. Thanks.
I'll just add that he puts his construction skills to good use around a lot.
Thank you for the kind words, Tim. Hello, everybody. I'd like to thank you for spending time out of your day to listen to me present on an interesting topic. Hopefully, I can offer insights for technology I believe will revolutionize several industries in the near future, including agriculture. Today, I'll be discussing the topic Marching to the Rhythm 10101, Artificial Intelligence as a Complement to Genomic Selection. We'll begin by discussing the pathogen Sclerotinia sclerotiorum, which is a devastating disease of many important crops, including soybeans. Control strategies for the pathogen include row spacing, reduced irrigation before and during flowering, or the use of biological control agents. Unfortunately, implementing these management strategies to control the disease are not very effective. Due to the lack of control strategies, the pathogen decimated 40 million bushels of soybeans in 2015. While fungicides are effective in controlling disease severity and preventing yield loss, profitability per acre does not increase. Currently, the best strategy for management of the pathogen lies within genetics of the host. Since resistance to the pathogen is found within the genetics of the host system, it is important for breeders and plant pathologists to coordinate on trait development. One of the oldest and most productive methods for providing disease resistance in a crop is traditional breeding. Since the majority of us are plant pathologists, I'll quickly remind us how traditional breeding methods select for resistance. The process begins by selecting a resistant cultivar and crossing that with a recurrent elite line, which is susceptible. The offspring in the first sexual event is again crossed with the recurrent parent. The progeny of the second cross is selected for the trait of interest and again crossed back with the recurrent line. At the fourth back-cross generation, the recurrent parent contains 96.9% of the DNA belonging to the superior line. The goal of back-crossing is to obtain genetics as identical to the recurrent parent, all while maintaining the gene of interest. Depending on the type of crop, this process can take 5 to 12 years. A more advanced form of breeding was developed known as marker-assisted selection. To reduce the time involved with breeding efforts, the simplest form of this approach is marker-assisted backcross. Similar to traditional breeding methods, you begin by crossing a resistant cultivar to a susceptible elite variety. However, the resistant variety will contain a molecular marker associated with the trait of interest. Breeders then cross the F1 progeny. After crossing the F1 progeny, the first backcross generation is genotyped during the seedling stage. If the seedlings express a polymorphic marker associated with the trait, the plants are selected for further backcrossing. Once the fourth backcross is complete, the genome will be 100% homozygous at the selected markers and contain a recovered genome of the elite parent up to 99%. One benefit of this breeding approach is the ability to purify the recurrent genome in a shorter amount of time, typically 5 to 10 years. Another benefit of marker-assisted breeding is that lower plant populations are required for the trait screening process. With the advent of next-generation sequencing, a faster, more refined version of marker-assisted selection has been developed, known as genomic selection. The goal of genomic selection is to predict breeding values of each individual without the need for directly identifying QTL regions. First, you begin by identifying a disease-resistant training population with sufficient pedigree information. Then, each accession of the population is genotyped using genome-wide association markers. The test population genotype is used to train statistical data for predicting traits. After the statistical parameters have been generated, test populations are genotyped in an early developmental stage. Using the genotype data from the breeding population, breeders calculate a genomic expected breeding value. Using pedigree information and genomic expected breeding values, breeders can combine and analyze this data using a neural network. Then the plants are selected for backcross. At this point, you're probably asking yourself, how are researchers able to identify disease resistance using markers? The resistance traits are characterized by genotype in the plant. Researchers determine single nucleotide polymorphisms occurring within each loci. These regions are known to breeders as quantitative trait loci, a section of DNA correlating to a variation in the phenotype of the plant. Now let's look at the chromosome on the left that contains 3 specific regions. M1 and M2 are molecular marker loci that are inserted to plant QTL. For the QTL to be detected, both QTL and a nearby marker must be polymorphic, which means the parents contain detectable differences in alleles at the loci. In this model, the M1 marker is considered polymorphic. However, the M2 would not be. At this point, you begin to screen for phenotypes within the F3 population that correspond to QTL regions. This figure is a representation of the breeding techniques previously discussed. All 3 approaches reach similar outcomes, but utilize different technologies to reduce time and inputs. Implementing genomic predictive models can reduce the time required at greenhouse and field screening steps, depending on the type of crop selection, and reduce the time required for trait breeding by up to 6 years. To assist with the selection process, researchers have begun to analyze data sets using machine learning algorithms. Both marker-assisted breeding and genomic selection techniques utilize the algorithms. However, genomic selection can employ a more powerful form of computational analysis known as artificial neural networking, which analyzes pedigree information, weather data, and genotypes to generate improved prediction accuracies. 2 characteristic differences between traditional breeding methods and marker-assisted selection are the genotype step and the downstream data analysis. While machine learning is certainly a form of AI, it is not what one imagines when thinking of AI. Machine learning utilizes various statistical methods to analyze vast sets of data known as the training set. The analysis uncovers patterns that can be used to accurately predict outcomes of future data sets. There are 2 varying components to machine learning algorithms: regression analysis and the statistical methods. Regression analysis takes large sets of data and interprets what is statistically significant within the data set. 2 methods most commonly used for regression analysis are ridge regression and least absolute shrinkage and selection operator. Statistical methods analyze this data, then predict outcomes based on that data. Once the prediction is made, it is cross-validated against known solutions, and the intrinsic parameters are adjusted accordingly. This process is reiterated using the training set until sufficient prediction accuracy is produced to warrant making predictions on data with unknown outcomes. 2 of the most commonly used statistical methods are BLUP and Bayesian inference. When conducting experiments, researchers can easily be swimming in a sea of data. Not all of this data is statistically relevant. As in any experiment, statistical outliers will exist. But in huge sets of data, how do we manage to accurately find the trends in the data by putting emphasis on the statistically relevant? This is where regression analysis is applied. Regression analysis is a set of statistical processes for estimating the relationships among variables. 2 of the biggest hurdles when statistically analyzing large data sets are models that either underfit or overfit the data. Let's use this data set as an example. Underfitting is when the statistical model is unable to distinguish the statistically relevant data points and treats each data point as the same. On the left is a line of best fit. However, you can visibly see the data points are drastically different. Next, we have overfitting, which occurs when the statistical model only recognizes statistically relevant data and ignores the rest. This ends up giving the same result as if you had just not entered the data. As you can imagine, this would lead to a particular solution that would be terrible at predicting future outcomes. What we are striving for is a best fit function. A best fit function will take into account each data point, but it will accurately weight each on its relevance. This is where regression analysis comes in. With it, we want to give the statistical model its best fit function so that it can give us the most accurate predictions possible given the initial data. The simplest example of regression analysis is linear regression. This is something that most people who have modeled data in an Excel graph have used. Whenever you create a graph in Excel and have it modeled a best fit line, the software is using linear regression to calculate the solution. Let's quickly go over the basics of linear regression and start with the equation of a line, where m is the slope and b is the y-intercept. Now, let's take a look at the linear regression equations. First, let's focus on the correlations between the vector form and the equation of a line. As you can see, the y and the x are in familiar places, but the other two variables are different. We have m and beta in similar spots, and b and epsilon. It is given that epsilon is our error term, which correlates to the intercept, but the only one we are interested in is beta, because it is the variable that we are trying to find when doing calculations. This is interesting because this means all we are doing in linear regression is solving for the slope within the error of epsilon. Finally, I'd like to quickly bring your attention to the summation form of the linear regression, which is the form the equation a computer would use to perform its predictions. Now let's talk about the regression methods commonly used in marker-assisted selection. These are ridge regression and lasso. Both of these methods were developed as a means to minimize the error that can occur in linear regression, which can easily be written out since there is a term in the equation. Given the vector form of the equation, we want to find the error in terms of the variable we are searching for, beta, which can be done with some simple algebra. Then, to find the total error, we simply take the magnitude of the vectors divided by n, which is the total number of input variables, and we have our average error function. In this equation, y is the output of the statistical method and x is the input to the system. So both of these are known. What is unknown is beta, as beta is what we're solving for. So if we want to adjust the error of the system, beta is the only variable we can manipulate. The approach is how this function is minimized is the only difference between these two methods. Since beta can be any vector of real numbers, the minimization problem posed is no easy task. To make this problem manageable, both methods impose constraints on beta. Ridge regression imposes the constraint that beta squared must be less than or equal to 2, where t is an arbitrary constant, while lasso imposes that beta must be less than or equal to t. This figure is a good graphical representation of what these constraints do to the method's ability for minimizing the error function. On the left, we have lasso, and on the right, we have ridge regression constraints mapped on a 2D plane. Beta hat represents the perfect solution for the minimization problem, which will realistically never be obtained. The red rings around beta hat represent how the regression solutions move away from the true minimum. It is where the red rings intersect the blue regions that is of interest, because this is where our regression formula intersects with the constraints of that the method is imposed. These will be the solutions that each method gives us, and as you can see, both methods will return similar results, but the edges of the regions are what make it so that they are notably different. The straight edges of lasso constraints means that often you will have zeros within your solutions, shown by the corners of the diamond. The logic behind the usefulness of this method is that coefficients are small enough, they will basically have no impact and be zeroed out. However, with ridge regression, we have smooth circular region, which means that the threshold for zeroing coefficients is much higher. Now that we've gone through the mathematics of these two commonly used regression methods, let's dissect what it all means. With ridge regression, the system is able to take into account smaller variations within the data. But how useful is that? Well, if there are minute correlations that would go unobserved, this would be extremely useful. However, much like humans, the system would also be reading into the data too much to try and find correlations that don't exist. On top of this, ridge regression is slightly more computationally expensive due to less zeroing. On the other hand, LASSO is less expensive computationally due to more zeroing of coefficients. This has both its pros and cons. Sometimes ignoring small correlations not only helps computationally, but can make your predictions more accurate than ridge regression that identify the useless correlation. That being said, it is easy to see how ignoring such correlations could also be a hindrance. Using our short crash course in regression analysis, let's examine a case study. For this study, the authors tested predictive models for disease resistance using two separate wheat lines developed by the International Maize and Wheat Improvement Center. The varieties developed at the center are referred to the 45th and 46th International Bred Wheat Screening nursery stock. The data network consisted of plants grown in fields across Mexico and several locations globally. What we want to focus on are the regression analysis approaches. Does ridge regression or LASSO provide better results for regression analysis? Also, are the predictive abilities of a least squared equivalent or better than machine learning methods? Our first comparison examines the ability of each approach to predict for Septoria-tritici resistance in wheat. As you can see, the machine learning methods significantly outperform the standard statistical tests, while there are no observable differences between ridge regression and LASSO. Here we are looking at Stagonospora nodorum resistance in wheat. The 45th year breeding stock shows statistical differences between the least squared approach versus the regression analysis. However, there were not differences in the 46th year breeding stock. This data suggests that predictive models for certain host pathogen interactions could have a higher degree of variability depending on the underlying genetics of the system. Prediction accuracies for the resistance to Frequentist or repentis in wheat were higher when analyzed using ridge regression and LASSO compared to the least squared approach. Contrary to the second example, the 46th breeding stock prediction accuracies remain statistically different than the least squared approach. Overall, the average increase in accuracy using genomic prediction models compared to the least squared approach was 48%. Now that we've examined the regression techniques to normalize the data, let's delve into the statistical prediction methods, which are the powerhouse of the analysis. Best linear unbiased prediction, or BLUP, is a statistical method for estimating random effects, such as a plant receiving a disease resistance trait. As the name suggests, BLUP is based on a linear model, similar to how we began linear regression. A graphical representation is shown here, with the distribution in light blue. The expectation values for each equation intersect at the distribution. A linear predictor is a function used to predict information given for a distribution, and any predictor that is worth using will pass through the center because the expected value for x should yield the expected value for y. So all of these lines could be considered linear predictors. What sets BLUP apart? BLUP is able to analyze for the means. BLUP is able to minimize the mean squared error. This can be thought of as a best fit line, as a line best fit to the contour of the distribution. See here how the blue line on the right follows the shape of distribution more so than the orange lines on the left. This line represents the BLUP prediction, whereas the others are random linear predictors. In the standard view of statistics, formerly known as the frequentist approach, prediction often involves finding an optimum point estimate of the parameters. Then the estimate is applied to the distribution formula to get the inferred data point. The major drawback to these common statistical methods is a tendency to underestimate the variance of the predicted distribution, which is caused by not accounting for uncertainty parameters. A common remedy for this problem is the frequentist approach, using confidence and prediction intervals constructed from a t-distribution. However, the Bayesian approach is much different. In the Bayesian model, you start with initial prior distribution, an objective distribution determined by your data space. From there, you cycle through the data sample, and using Bayes' theorem, you essentially apply the knowledge from the data point to the distribution. After this, you are left with a probability distribution that has learned from the data and is ready to make predictions. Before we jump into Bayes' theorem, let's quickly review some terminology. Here we have the conditional probability of A given B. This is the likelihood that A will be true if at first you know that B is true. Bayes' theorem is most often seen as a framework in frequentist methods for simply finding conditional probabilities based on the available information. However, we can find another interpretation using it as a model for inference and laying the groundwork for Bayesian probability. If we want to know the probability of A with respect to B, it is obvious that we want to know the probability of A before accounting for B. Thus, we call probability of A the prior, because it is the probability prior to taking B into account. The probability of A before B is known as the posterior because it is the probability of the A after accounting for B. Finally, the probability of B before A over the probability of B is the evidence that B provides for A. Predictive algorithms are only as good as their predictive distributions. The key to a good predictive algorithm is finding the most accurate distribution possible. Bayesian prediction accomplishes this by making the parameters of the distribution be the factor determined probabilistically rather than the data. This infographic illustrates how parameters are used in Bayesian to simulate, in some sense, thinking. At the heart of all AI operations is simply data analysis, which is also the basis of intelligence in general. Let's look at how the Bayesian model handles data. First, the objective prior distribution is generated based on the magnitude of data points. Then the model cycles through the data set, applying Bayes' rule on the distribution. Using each data point to refine the parameters and overall distribution accuracy, the continued application of Bayes' rule until certain error conditions are met is known as training the model. After this process, we have a posterior probability distribution along with a set of parameters that have been trained to the sample. From here, we can integrate the posterior distribution over the parameters to predict the outcome of the data points that aren't in the training sample. This training method is only valid for behavioral data that follows a pattern of natural behavior and not just any random data set. It is this way because the parameters can be thought of as mental constructs, essentially placeholders for the model to construct a pattern in the behavior. For our second case study, researchers began by screening the USDA germplasm for resistance to Sclerotinia sclerotiorum. The authors identified 19,642 accessions that exhibited white mold resistance. After the initial screening phase, 465 of those soybean lines were selected from maturity groups 1, 2, and 3 for further analysis. 352 of those accessions were selected to train the genomic model, while the remaining 88 were used for validation of accuracy. Genomic expected breeding values were calculated for each genotype, and the prediction model was selected based on the highest accuracy. The control set selected the superior 5 and 10% genotypes, as well as the inferior 5 and 10%. The testing set used previously reported studies on white mold resistance and chose 28 soybean accessions for determining how well the model could identify sources of resistance. Genetic expectation breeding values were calculated for each set and analyzed using the predictive method. The box plot here is displaying comparisons between the control set as well as the test set. As you can see, the predictive accuracy of the test set aligns with the top 10 accessions exhibiting white mold resistance. Highlighted in red is the testing set. When comparing the differences in accuracy between predictive models, researchers observed no statistical differences between BLUP and Bayesian. However, it was noted that BLUP reports slightly higher prediction accuracies in greenhouse trials when compared to Bayesian networks. Genomic prediction models are often utilized in greenhouse environments to infer phenotype drift. field setting. To determine if this approach is accurate for field prediction models, the authors wanted to cross-validate prediction accuracies between the two environments. The data suggests that the development of white mold in the greenhouse setting is controlled by a different set of genetics when compared to field setting. Therefore, proper considerations must be made when regarding the type of host pathogen interaction in order to select the correct genomic prediction analysis, models for analysis. Now that we've analyzed two of the most commonly used statistical methods for machine learning algorithms, BLUP and Bayesian, let's compare the predictive accuracies of an artificial network to the Bayesian model. Before going into the literature, I want to refresh our minds in regards to the biology of a neuron. After all, the key concept of neural networking is neuron connectivity. Initially, our dendrites flex electrical signals, shown in green. Then the cell body will integrate these signals to generate an outgoing signal to the associated axon, shown in gray. Once the axon receives the electrified signal, it will pass to the linked dendrites or an effector cell, blue. These vast networks of neurons and the excitations that occur allow us to perceive and experience life. Each individual neuron has a part in the perception of the world around us, but it is the network as a whole, the construct we call a brain, that gives way to the levels of intelligence we are able to achieve. Our ability to perceive and learn from the world is controlled by molecular mechanisms governing neuron regulation. As children, we utilize our neurons to learn from our surroundings. Each and every one of us successfully hone this learning method by using a process similar to the scientific method. Artificial neural networks are capable of replicating a process similar to humans. Initially, we are presented with our problem, such as Sclerotinia sclerosis. From the problem, we develop a purpose for addressing the issue. In our case, improving the genetics of the host system. Next, we hypothesize an outcome. Let's hypothesize that artificial neural networks will perform worse than Bayesian models since they're fairly a new concept. Then we develop the experiment to test the hypothesis, comparing Bayesian versus artificial intelligence. After obtaining the results from the experiment, we can analyze the data, which will be terabytes of genotype data, pedigree information, and environmental data. Finally, we reach a potential conclusion that will hopefully help us increase yield. Let's look at an example for a visual representation regarding the learning process achieved using artificial neural networks. In this example, we want to teach the neural network to identify pictures of an animal and distinguish what it could be. During the training phase, a neural network is fed thousands of labeled images of various animals and learns to classify them. For the input phase, an unlabeled image is shown to the trained network. At the first layer, the neurons respond to different simple shapes, such as edges. Once the analysis reaches the higher layer, neurons can respond to more complex structures, such as a tail or nose. At the top layer, the neurons respond to highly complex, abstract concepts that humans would identify as different animals. Finally, at the output stage, the network is trained well enough that it is capable of predicting the object based on the training set. We can now apply our current understanding of neural networking to our issue with pathogen Sclerotinia sclerosis. To help us solve our issue, we will use a conceptual neural network. Unlike a biological brain, where any neuron can connect to any other neuron, artificial neural networks have discrete layers and connections that dictate the direction of data propagation. Initially, we have our observations, which can be thought of as listening with the ear or seeing with the eyes. Then the neurons in your brain begin to think about the observations. A prediction can be made, and we are ready to conduct experiments. An experiment could look at how many isolates of Sclerotinia sclerosum were resistant to a particular fungicide, or perhaps the soybean variety that exhibited resistance to the pathogen, or the amount of increase associated with a particular management strategy. Once the experiment is complete, you analyze the data. Then the findings are reported back to your neuron nodes. And by reiterating this process over and over, the system will eventually begin to learn from the data set, similar to a human. Since computers are currently incapable of interpreting language, we are unable to fully implement the scientific method as a thinking process. So we must revert to mathematics in order for the system to understand the input. We can think of the real population as a vector. So each real population is a vector of several reals. Each reel is a vector of a bunch of SNPs. Using those SNPs, we can multiply them to weighted values present to a particular neuron or node. After multiplying the weight, an output is expressed as alpha. This data will then be fed through a filter or a hyperbolic tangent, allowing for the data to become linearly separable. You can then repeat the procedure for each neuron as many iterations as possible. The higher amount of layers within the system can create more dimensions of complexity. Increased complexity allows you to conduct logic gates. If this SNP and that SNP, then what is the result? Once it is analyzed by the neuron layers, you will receive an output expressed as Y. Then you conduct an experiment to determine the population of spores that were resistant to the fungicide and compare against output Y, which is your air value. Using the air value, we can report the finding by dividing the change in air over the change in weights. To utilize GPU power, the equation must be converted to linear algebra using the chain rule. Now we have a formula allowing us to report the weights back to the system, which is done one at a time. We complete this process over and over until eventually the system learns from the data and minimizes the error based on each cycle. The math is over, I promise. The final case study explores the use of artificial intelligence versus predicting Hymelia vastrix resistance in coffee arabica. One downfall of BLUP or Bayesian models are the inadequacies for determining disease resistance characterized by categorical variables. To overcome the pitfall of Bayesian networks, Silva et al. Developed a simple neural network containing one hidden layer related to 137 molecular markers, directly correlated to resistance. The neural network created by Silva contained a total of 6 neurons with a maximum number of iterations equal to 5,000. Surprisingly, during the training population, both approaches obtained an error rate of 0%. However, during the validation step, artificial neural networks obtained a classification score with error rates of 1.6% compared to the Bayesian model of 2.4%. The overall agreement between the two approaches was 81.37%. For cross-validation of the model, the authors examined the efficiency of the artificial neural network's ability to predict resistance using a limited number of markers. It was conducted that artificial neural networks perform best using a large amount of information, such as pedigree, genomic expected breeding values, weather data, and even environmental factors, including soil type. So in conclusion, artificial neural networks are an interesting and promising approach for predicting plant disease resistance via genomic selection. While artificial neural networking is still in its infancy, the models can still be used to evaluate and predict disease resistance in plant populations more efficiently than Bayesian network approaches. If Moore's law continues at its current rate, several advancements, including improved next-generation sequencing platforms, increased computational processing power, and more advanced artificial networking algorithms will only improve the accuracy of plant disease resistance inference models. Thank you. Dr. Vincelli?
Yeah, thanks for bringing something out of the ordinary to the seminar series. Yeah, it's good to hear that. So all of the techniques you described, if I'm not mistaken, are sort of like correlated. In other words, associate this marker with that with resistance. Am I correct? Those are not mechanistic. There's no mechanistic knowledge necessary, but these are correlation processes that are used to evaluate. Is that right?
Yeah, that's correct. The only difference would be the amount of time that it would take for each process, which, depending on the type of crop, can be expedited up to 6 years with genomic selection when compared to traditional breeding methods.
Back to your slide with the greenhouse and the field, predictive values for the greenhouse and the field.
The box plot.
And there were two. Yeah, there. Next one. So I guess we're looking at predictive accuracies. What is the percent value actually telling us?
How well that model predicted disease resistance in an unknown accession line. So they used those 28 accessions from they were characterized accessions, but they just used those as like to check the model's accuracy.
So these aren't progeny from a breeding scheme.
No, these were pulled from literature. So it was trained using the accessions that they had picked from the USDA database, and then it predicted how the disease resistance from the literature.
Basically, like it's a whole association.
Yeah.
But going back in and refining the model,
Mm-hmm.
that's the AI part.
The AI portion of it is simply just feeding all that data back into like what would be a node. So you would have one node, another node, kind of like that picture that I showed you, similar to that.
Kind of on the same line of question, assuming this is quantitative resistance we're looking at, not qualitative. So if
No, I'm sorry, it's both, because they do phenotype screening once they go out into the field. So they initially do the genotype screening. From that, they plant those genotypes in the field, give those a expected breeding value based on the phenotype that's shown under disease pressure. And then that value is also inputted.
So I guess looking at your y-axis and kind of stepping back, you're not the researcher who generated this data. Is a 15% success rate, do you consider that good?
No, I would not. I would not consider that good.
But okay, that wasn't a trick. But then the real question is, how would you improve the model? What are some suggestions you have on improving the prediction accuracy of that model?
I think it really just comes down to pathogen host genetics. Like, for example, this system would thrive on humidity, and you would have a lot higher humidity in a greenhouse as compared to some field settings, especially for Sclerotinia, which is grown more in northern locations.
I guess overall, that wasn't really the question. The question was, what are some other variables that they haven't already incorporated in models that can capture more of that variation? Clearly not.
So they didn't include environmental parameters. This was just pedigree information and the genotype data. That was just, yep, just pedigree and then genotype data based on their sequencing. They actually looked at that in the study and said that the number of plants, once you reach a certain population, that it's just a diminishing return.
So with these data, if you look on the left, you've field predictive accuracies. That's not very accurate either.
No.
So this is before you go into the neural.
This is before. This is using BLUP and Bayesian networks.
Did you show us what you get if you look at these data sets using?
They don't analyze it using neural networks, unfortunately. Neural networks they're actually a pretty old concept. They were first developed in the 70s, but we didn't really have the computational power at the time to utilize them. So now we're kind of revisiting them, and it's kind of like a rebirth, and everybody's trying to figure out algorithms that actually utilize them.
The message from this slide is really they're not very good. You wouldn't even be able to apply these to breeding.
No.
Maybe the neural network.
Yes.